So it is my pleasure now to introduce Dr. Drew Weissman. Dr. Weissman is one of the recipients of this year's Canada Gairdner International Award for his pioneering work with Dr. Carrico in developing nucleoside-modified mRNA therapeutics, of course, the foundational technology for the highly effective COVID-19 mRNA vaccines that we've been hearing about. He is the Roberts Family Professor in Vaccine Research, Director of the Penn Institute for RNA Innovation, and Director of Vaccine Research, Infectious Diseases Division at the University of Pennsylvania, Perelman School of Medicine. So I am delighted to turn it over to Dr. Drew Weissman. Thank you.
Thank you very much. So I'm highly honored to receive this award. We've been treated great in Canada, visiting all ends of it, from the sounds of it, of all the other awardees. What I wanted to do in today's talk is to give you an idea of the potential breadth of RNA technologies in the future. Hopefully everybody in this room has received a vaccine in their arm, but there's huge potential for what RNA can do in the future. Let me just start by telling you the difference between RNA vaccines and all the other vaccines made. Making an RNA vaccine is very simple. You design the sequence, Katie described this. You in vitro transcribe it, that's a 2-hour reaction. You purify away the contaminants. You mix them with lipids that forms nanoparticles. You get rid of the ethanol and salts, and you put it in a vial, and you've made a vaccine. If you want to change the vaccine, you change the sequence design. Everything else is identical. For all other vaccines, when you change the vaccine, when you make a new vaccine, you start over. You figure out how to produce the protein, how to purify it. Everything is different. Everything is a huge amount of work. So when we started looking at vaccines, we looked at influenza. This is just a review of the current literature when we were first investigating influenza vaccines. The first two are similar to the human vaccines, the live virus we give to kids, the inactivated virus that we give to adults. And what's in parentheses are the viral titers you see in mice after 2 immunizations. It's called an HAI titer. It's a pseudo-neutralization titer. But you can see with all of these, titers in the 1 to 100 range are typical. 1 to 40 is protective, so this is considered a good titer. When we looked at HA encoding mRNA, and we're now comparing it to a live virus or inactivated virus, the titer from mRNA was 1 to 2,500. That's 50 times better than an activated virus, 5 times better than live virus. This excited a lot of biotech and pharmaceutical companies, because typically in vaccine research, if you can double a titer with a new vaccine, people are excited. But as an immunologist, we wanted to understand why this vaccine was working so well. So this is a cartoon of how a B cell response develops. Follicular B cells interact with particles, viruses, antigens, adjuvants. They get help from CD4 positive T cells and form germinal center B cells. The germinal center is the site where the B cells rapidly proliferate, avidity mature, and form plasma cells and memory cells. So we measured all of these populations. We're measuring the number of antigen-specific B cells of each type. We have a fluorescinated HA in the flow cocktail, and then other antibodies that identify each of these 3 populations. What you can see is that in all of them, we have about a 50-fold increase in the number of antigen-specific B cells. That accounts for the increase in antibody titer. We left some mice for over a year and went back and we measured the long-lived plasma cells in their bone marrow. Again, we saw huge numbers of these cells still present after a single immunization. But that didn't explain the mechanism. And we wanted to understand why we were getting these huge antigen-specific B-cell responses. It came down to what are known as T follicular helper cells. These are a form of CD4 helpers that form germinal centers, and they supply the help to the germinal center B cells in order for them to affinity mature, proliferate, and make memory and plasma cells. We used a macaque model system, and in these macaques, we gave them either an mRNA that encoded the HIV envelope, or we gave them that same envelope protein with a double-stranded RNA adjuvant. We chose that because that was one of the best described adjuvants for inducing TFH cells. But as you can see, there's no comparison to the effect of the mRNA-LNPs and the double-stranded RNA adjuvant. Huge numbers of TFHs were induced by the mRNA-LNPs. We did a calculation. Typically, for most vaccine adjuvants, double-stranded RNA, alum, MF59, 5 to 10% of the CD4 helpers are TFH phenotype. For mRNA-LNPs, it's over 50%. So this is an adjuvant that selectively induces TFH cells that drives B-cell responses. That's what accounts for the huge antibody responses. We wanted to test these in an animal system. So in influenza, every year you have to make a new vaccine because the head of the virus, the HA, mutates and prior vaccines don't work. We challenged the animals with a different subtype of HA. This is from an H5 avian virus that occasionally causes pandemics. We saw complete protection with an H1 vaccine against an H5 virus. This is now going forward into humans as a universal influenza vaccine. I want to switch topics and now talk about the future of RNA therapeutics. If you've worked in this field, you know that the critical difficulty is targeting, is getting the nucleic acid or the virus to the cells of interest. With LNPs, they go to the liver. They bind ApoE and they target hepatocytes. They also go to dendritic cells, which makes them a good vaccine. We wanted to know, could we target LNPs to other cell types? We concentrated on endothelial cells, T cells, and bone marrow stem cells. The platform that we used was to simply change the surface of the lipid nanoparticle. We attached antibodies, pieces of antibodies, receptor ligands, other proteins to the surface of the lipid nanoparticle using different types of conjugation chemistry. When we did that, we saw that we didn't change the morphology of the LNPs. They got a little bit bigger, which is the addition of an antibody coating, but they retained their surface charge. And most importantly, they retained function. So you can see in C, these are UVECs, primary human vascular endothelial cells. In the absence of targeting, you get no delivery of LNPs or mRNA. With targeting, we're using a PCAM antibody to target, and PCAM is expressed by UVECs. We see high levels of uptake and translation of the mRNA. So we could selectively target human endothelial cells in culture by targeting them with a PCAM antibody. We injected these LNPs into mice, and what you can see are really two things. The first is that when we added an antibody to the surface of the LNP, we reduced liver uptake. We showed that we're actually blocking ApoE binding. Most importantly, we're now seeing lung uptake, and about 50% of the delivered LNPs are going to the lungs. Lungs have the highest level of PCAM on endothelial cells. We wanted to see what else could we use this for. So in the cerebrovascular elements of science, strokes are a common occurrence. Typically, when a patient has a stroke and you make a CAT scan 24 hours later, you see a small area of dead brain that's fed by that middle cerebral artery that had the stroke. But that's not the main problem of a stroke. What happens in the following days is the stroke extends and gets much larger. And that's because that initial stroke leads to edema, inflammation, and extension of the area of brain death. So we developed a system where we could inject TNF-alpha, a pro-inflammatory cytokine, into one hemisphere of a mouse brain. And then we treated those TNF-inflamed brains with a nanoparticle that targeted the brain, targeted the blood-brain barrier, using VCAM targeting. We delivered an mRNA that encoded thrombomodulin, which is an anti-clotting, anti-inflammatory protein. And what you can see on the left is that when we treated animals with a VCAM-targeted LNP with thrombomodulin, we got rid of all of the vascular leakage. So that in theory, would get rid of that extension of the infarct to greater portions of the brain as a potential therapeutic. My lab has had an interest in HIV for a long time. So we wanted to see, could we target T cells? We did the same thing. We added antibodies to the surface, and we could successfully target primary CD4 positive T cells. But most importantly, if you've ever worked with T cells, they don't have endocytic activity. That means they can't endocytose free particles. You can't deliver nucleic acids with lipids, polycations, or other particle delivery systems. The only way you can do it was electroporation. Our hypothesis was, if we had an antibody that bound an antigen on T cells that was endocytosed after binding, that would bring the lipid nanoparticle into the T cell. And that's what we're seeing here. Purified human T cells, targeted with ibilizumab, which is a clinically used anti-CD4 antibody, gives high levels of luciferase activity. The T cells are taking up the LNPs, the RNA is released, and it's translated. When we inject these into animals, the spleen lights up. But most importantly, we re-imaged those animals after we took out all of those organs. And what we're seeing now are lymph nodes. These are para-aortic and inguinal lymph nodes are lighting up. So the LNPs injected intravenously are getting out of the circulation, into the tissues, into the lymphatics, to the draining lymph nodes, and they still have the capacity to bind T cells, deliver the mRNA, which is translated and shows up. We switched to a model system that has a LOXP reporter gene in it, in this case, a green fluorescent protein. The mRNA that we delivered with the LNPs encoded Cre recombinase. So the Cre recombinase removes the stop codon and makes targeted cells green. We injected the targeted Cre recombinase LNPs into an animal, and without targeting, you get very little green protein. With CD4 targeting, you get high levels of green protein in lymph nodes and the spleen. But what's important to notice here is that on the y-axis, that's an activation marker. So we're seeing equal amounts of delivery of LNPs to resting T cells as we see with activated T cells. That's important because in HIV, the resting CD4 positive T cells are the site of HIV latency, and they have to be targeted in an animal or in a human. We also wondered, could we deliver CAR T cells? So this is what a CAR T cell is. CD8 positive T cells recognize antigen plus MHC. And when they recognize that peptide MHC, they kill those antigens on a tumor cell or a virally infected cell. An alternative was developed known as a chimeric antigen receptor. And what happens there is that the CARs recognize a specific antigen. And the first that were developed were CD19. CD19 is expressed by leukemias and lymphomas, and CARs that recognize CD19 delete the tumors. They have clinical efficacy in the 60, 70, 80% range. That's a huge advance in cancer chemotherapy using the delivery of CAR T cell. The problem is, this is what it takes to make a CAR T cell. You have to take cells out of a patient, you infect them with a lentivirus, you expand them for a couple weeks, and then you give cells back to the patient. It works great, but it costs $500,000 per dose, which is problematic. What we proposed is whether we could make CAR T cells in vivo. So our idea was that if we could target T cells, and in this case, we used a CD5 targeting molecule that targets both CD4s and CD8s, and encode the chimeric antigen receptor in the mRNA. The T cell would take up the LNPs, the RNA would make CARs on the surface, and we chose a CAR that targeted fibroblasts. We tested this in vitro. 80% of the T cells made the CAR. In vivo, 25% of the T cells express CARs on their surface. And we used a model of fibrosis. So if you make a mouse hypertensive, they develop cardiac fibrosis. Most cardiac diseases are mediated by the fibrosis that forms after the disease insult. This is an echocardiography, a way of taking pictures of the function of the heart. And we can see on the right, we're measuring diastolic and systolic volumes. When the heart becomes fibrosed, it can't function, and the volumes get bigger. In the bottom right is the ejection fraction. This is the amount of blood that the heart pumps out with each beat. When the heart becomes fibrotic, the amount of blood that's released is reduced, and the ejection fraction is reduced. So gray is a normal animal, yellow is a fibrotic animal, red is those animals treated with one treatment of CD5 targeted LNPs that make FAP CARs. And we can see we return their cardiac function to normal. When we stain the hearts of these animals, the normal saline treated on the right, the middle are fibrotic animals. You see high levels of fibrosis. The left is one treatment with FAP CAR LNPs targeted to T cells, and we return the fibrosis to nearly normal. So with an off-the-shelf mRNA-LNP, we could cure cardiac fibrosis in this animal model. Another thing we were interested was targeting stem cells. So bone marrow stem cells are the source of all of our red cells, white cells, granulocytes that allow our immune systems and our respiratory systems to function. Sickle cell anemia is a disease that has 300,000 people born with it every year. They're mainly in Africa, but you see them across the world. The problem is you can't do bone marrow biopsies and viral infections on 300,000 people a year in regions like Africa and India and the Middle East. It costs about $1 million per treatment, and the facilities to do these things aren't available. So our idea was that if we could find antigens that selectively targeted bone marrow stem cells, we could deliver LNPs and target those. And that's what we did here. We saw that with a single treatment, 85 to 95% of bone marrow stem cells and progenitor cells were targeted by the LNPs, and we could mediate gene recombination in these cells. Most importantly, what you need to do is called a secondary transplant. So you treat mice once with the LNPs, you wait 6 months and take their bone marrow and put them into irradiated mice that no longer have bone marrow stem cells. And we found that our targeted LNPs from the previous mice, 100% of the red cells, white cells, and granulocytes were gene recombined. So that means that with a single injection, and in this case it's 0.1 micrograms of mRNA-LNPs, could gene edit 100% of the repopulating bone marrow stem cells. So now we can go to Africa, India, the Middle East, the United States, Europe, anywhere there are people with sickle cell anemia. We can give them a single injection of mRNA-LNPs that target the bone marrow and that can correct the sickle cell gene defect and cure their disease. So instead of $1 million per treatment, now we're looking at, I don't know, a couple $100, maybe $1,000 per treatment. And we can easily cure sickle cell anemia with a single injection intravenously of RNA-LNPs by targeting them to the bone marrow stem cells and delivering an enzyme through RNA that fixes the gene defects. You can do a lot of other things. You can deliver toxic genes to stem cells. So now when we do bone marrow transplants, you have to irradiate people or give them high doses of chemotherapy to kill the bone marrow stem cells. The problem is it also kills the gastrointestinal cells and many other necessary cells. That has toxicities and mortality up to about 5%. Now we can deliver an mRNA-LNP. We can selectively kill the bone marrow stem cells and easily do transplants. We can also deliver therapeutic proteins that stimulate B cells, granulocytes, platelets, many other cell types that can be easily delivered to the bone marrow. So our conclusion are that mRNA-LNP therapeutics has the potential to do many different things. We have vaccines for many different diseases in clinical trials. We're developing ways of doing gene therapy in bone marrow stem cells. We can target heart, we can target brain, we can target lungs. That allows us to deliver mRNA-LNP therapeutics to many different organs and cell types. And we see this as the future of mRNA-LNP therapeutics. I need to thank all of the people in my lab and all of the labs that we collaborate to do these. That includes Katie, who together we developed nucleoside-modified mRNA that led to the vaccines for COVID currently in use. It includes Acuitas Therapeutics, which is a company that Peter Cullis started and who we've worked with for 10 years or so to develop LNPs as an mRNA delivery platform, and my other collaborators for all of these studies. Thank you.